At Pfizer, we've been committed to the rare disease community for over 30 years. Gene therapy has the potential to be a transformational treatment for patients, and we're committed to advancing this potentially life-changing approach. To do this, we've made investments in innovative gene therapy technology and facilities over the past several years, giving us end-to-end capabilities and the ability to guide a gene therapy medicine through its entire life cycle. Let's take a tour of our facility here in Sanford, North Carolina, to learn about this exciting technology, our manufacturing process, and how we've adjusted the process for global supply. There are several types of gene therapies. We focus on potentially one-time treatments that use a highly specialized viral vector to deliver a functioning gene directly to targeted cells. This functioning gene serves as a blueprint for the tissue to create the missing or non-functional protein that is causing a patient's disease. There are 4 steps to the gene therapy manufacturing process: raw material preparation, upstream processes, downstream processes, and product completion and packaging. Raw material preparation is the first step of manufacturing gene therapies. This is where we secure the different elements that are needed to manufacture the shell, called a capsid, that houses the functioning gene that will be delivered to the affected tissue. After raw material preparation, we transition to what we call upstream processes. This is where the newly created capsids containing the essential building blocks to create the functioning gene are encapsulated into the viral vector that will deliver it within the body.
The upstream process starts with a single vial containing HEC-293 cells. The contents of that vial is transferred to a tea flask that contains a media suitable for its growth. Once the contents are in the flask, it's placed in a 37-degree incubator and it incubates for 4 days. After 4 days, the tea flask is removed from the incubator and we check it for confluence under a microscope. Confluence is basically an estimation of how much of the surface area of the flask is covered with cells that are expanding. After several weeks, the flasks have grown from 1 to 3 to 10 to 26 flasks, and the cells are now able to be passaged to roller bottles. Once the production roller bottles have met confluence, it's time to start transfection.
After upstream processes are completed, the next phase is downstream processes. This is the purification stage where the non-essential cellular material is removed from the desired vectors. After the gene therapy is purified, we confirm the vector is working as intended and measure the concentration of the vector to ensure it's the perfect volume.
The process is executed by highly skilled manufacturing technicians, combining both manual and automated operations. Since the process is run in single-use equipment, many of the highly customized manifolds that are used utilize pinch clamps that need to be manually manipulated, whereas larger scale processes run in stainless steel equipment would typically utilize automatic valves. While portions of the processing are automated, like the chromatography operations, a large number of connections, pinch clamp manipulations, and operations are manual and depend on the flawless execution of our detailed procedures.
Once the downstream process checks and tests are complete, we're ready to package the gene therapy for clinical or commercial use.
The filling process begins with pooling of the drug substance, and then we filter the drug substance. Once the product's sterile filtered, we bring it into the drug product suite and we connect it to the back of the isolator. The isolator has gloves where we can manipulate the process, and we connect a sterile filling needle up to a robot, and then we deliver the drug through a pump through the filling needle, and then the robot does a large majority of the work for us. So it's an automated robot, It fills each vial in what we call nests until that nest is full. And then we run a laser across each vial to seal the opening that was created when the needle punctured it. And then we cap each vial. And all that, again, is handled by a robot. The end process for a drug product is to prepare the vials, or label them, and do what we call primary packaging and send them off to a clinic for CTM, clinical trial material, which is the campaigns that we're doing at the moment.
We also have 3 secondary packaging sites, one in the US, one in Japan, and one in Europe. Our European site is based in Spain and has the ability to pack and distribute direct to clinics in Europe, the Asia-Pacific region, and the rest of the world. Manufacturing gene therapies is challenging and resource-consuming, with an average manufacturing process from start to finish of approximately 9 to 10 months. Production is complex, requiring specialized technology and high levels of expertise. Over the past few years, we have made investments in our gene therapy manufacturing and distribution technologies to support the commercial scale manufacturing of our 3 Phase 3 gene therapy assets. At Pfizer, we're proud of the work we're doing to advance this exciting science. With a shared urgency, passion, and commitment, we hope to unlock the potential of using genes as medicine to make breakthroughs that change patients' lives, today and in the future.